We're now going to talk about the discussion section, particularly how the discussion section tries to put all the data that it just discussed in context with what was discussed in the introduction. So, what should be found in the discussion section? First, a review of the conclusions of the data, so the interpretations of the data that the authors discussed in the results section. And then explain how the data support, or do not support for that matter, the central hypothesis of the research article. Presumably, determining if the hypothesis is supported or not supported will help to fill gaps in the knowledge in the field, and then also relate the current study to the broader relevance of the field. So the discussion section not only discusses the data within, but may also draw from conclusions from other papers or critical evaluation of other papers that are discussed in the field in order to make the case for the author's conclusions. So we're going to go back to Thomas et al. So this paper, okay, and look at the discussion section, sort of analyze this. So here's the discussion section of this paper. So the discussion section starts with peroxynitrite is often presented as the sole source of nitrotyrosine in vivo. So this is recalling a statement that was discussed in the introduction, which is color-coded here in yellow. So detection of nitrotyrosine in vitro following exposure to synthetic peroxynitrite initially suggested that the reaction of NO with superoxide may be the primary source of nitrotyrosine. So this is indicating what one of the beliefs in the field is that peroxynitrite is largely responsible for nitrating tyrosine residues. Okay, and so this is also supported by data that is presented in this within this paper in Figure 1. Okay, in the next sentence that's highlighted in pink, it discusses data in Figure 2, discussing the complexity of chemistry resulting from simultaneous generation of nitric oxide and superoxide. It's emphasized by the dramatic reduction in NT yield at non-stoichiometric reaction amounts. So this is just discussing the description of the data in Figure 2a as well as their interpretation. And so the interpretation goes on further to echo what was discussed in the intro. So the dramatic decrease in yield of nitrated protein observed when the concentration of NO or superoxide is in excess demonstrates the requirement for specific conditions and reactant fluxes of nitric oxide and superoxide. Okay, so this data is discussed in Figure 2a, and this statement is made, which echoes a sentence that was also made in the introduction and color-coded in green as well, where these two sentences discuss the complexity or disparities in different results within the literature, particularly those where peroxynitrite are either made with nitric oxide and superoxide or reacted in the presence with nitric oxide or superoxide, and discussing the complexities of these reactions. And so the point of this sentence highlighted in green is intentionally to echo back to the introduction and say, well, these data appear to explain why some of these results are hard to interpret or are inconsistent across other studies. Okay, so remember, part of the point of this, of discussing this in the introduction, as we discussed in a previous video, is that they're making the case in the introduction that there are alternative pathways towards nitrating tyrosines. Okay, so in this case, that's what they say exactly in the sentence, is that there is evidence for an alternative mechanism through peroxidases. So they say here, thus provide an alternate mechanism for its formation in vivo, and that leads to what we identified as sort of a maybe hidden hypothesis statement within this third paragraph, where the authors say this may result from variations in protein susceptibility to nitration due to association with redox active metals. So essentially saying that redox active metals may play a huge role in tyrosine nitration. Okay, so that was our hypothesis statement. So in the discussion from this paragraph we just analyzed, the authors continue to talk about their data, largely which is looking at testing the hypothesis how metals may be involved in nitrating tyrosine residues. So they even restate this idea, restate the central hypothesis here, where they say, given the challenge, even our ideal conditions, it seems unlikely that in the complex setting that peroxynitrite should be considered the major source of nitrotyrosine in vivo, and alternative biochemical pathways become necessary. So this is a restatement of the hypothesis statement that we identified. And then from there, the authors in several places that are highlighted in green here present evidence such as figure 3 here, figure 4 and 5, figure 7, various ways in which they show that metal helps to cause nitration of tyrosines. And so a lot of this, the rest of the discussion continues to provide supporting evidence for this hypothesis. And so this is an interesting set that I want to highlight, where the authors say iron and copper levels have been measured as high as 0.4 and 1.0 millimolar, respectively, in the brains of Alzheimer's patients. NT formation on BSA in the presence of nitrite and peroxide was catalyzed by free ferrous, ferric, and cupric ion, figure 7. So the authors are continuing to make the case that nitrotyrosine increases in the presence of metals, and they weave not only their own data from figure 7 in this particular part of the argument, but also references from the literature where they discuss metal concentrations are higher in places where you might see nitrotyrosine residues, such as in onset of Alzheimer's. And so that is the goal. So one thing that is useful in reading discussions is to see how the authors view their data back into the broader context of the larger field, and watching and looking at the arguments made by both data and also previous literature in order to make this argument.